Welcome back, guys, to Kerstin's Microverse, where we talk about the fascinating and sometimes mind-blowing world of cell biology. Today's topic is one that is incredibly important to me personally, and I'm guessing to many of you too. We're talking about the connection between the cell cycle and cancer. I know, the cell cycle, it sounds like a snooze fest, right? But trust me though, it's anything but boring when you realize this tiny, intricate process in our cells is at the heart of diseases that impact so many lives. Now, here's where it gets a little bit personal. My guest today is me, yours truly, and honestly, I couldn't think of anyone better to tackle this. Why? Because this topic hits home. My mom has breast cancer, and I know you might know someone, or maybe even are someone facing this disease. I wanted to understand more. What's actually happening in her body? What does it mean when the doctors say her cells are growing uncontrollably? Why did this happen? I'll admit, learning about the cell cycle might sound super technical, but stick with me. We're going to break it down together. No PhD required, right? So my hope is that by the end of this episode, you'll not only understand what's happening at a cellular level when cancer takes a hold, but also studying why this process is key to finding better treatments and hopefully one day maybe even a cure. So, let's nerd out, get a little emotional, and maybe even find some hope as we explore the wild world of cell cycles, checkpoints, and cancer. Ready? Let's do this! Okay guys, before we start talking about the cell cycle and how it connects to cancer, let's start with the basics. What even is cancer? At its core, cancer isn't just one disease. It's a collection of over 100 different diseases. What all cancers have in common is this: cells in the body begin to grow uncontrollably. So, normally, our cells have a built-in system of checks and balances. They grow, divide, and eventually die in orderly fashion. But in cancer, this system breaks down. It's like a runaway train with no brakes. Cells just keep dividing and growing, forming masses called tumors. All right, guys, I hope you are keeping up with me. So, now not all tumors are cancerous. Some are benign, meaning they do not spread to other parts of the body. But malignant tumors, those are the dangerous ones. They invade surrounding tissues and travel to other areas of the body in a process called metastasis. Now, different types of cancer arise depending on where this uncontrolled growth starts. For example, carcinomas form in the skin or the tissues lining organs, like breast cancer or lung cancer. Sarcomas develop in bone, muscle, or connective tissue. Leukemia doesn't form solid tumors, but instead affects the blood and bone marrow. And then there are lymphomas, which start in the lymphatic system. The thing that makes cancer so challenging to treat is how it hijacks our normal biology. Each type of cancer has its own unique fingerprint, but the root cause is almost always tied back to one thing: the cell cycle. All right, so we know cancer is about uncontrolled cell growth, but let's dig deeper. How does it actually start? Here's where cell cycle comes into play. Remember, the cell cycle is like a factory assembly line, which each phase carefully orchestrated. cells only divide when absolutely necessary, like when you're healing from a cut or when new cells are needed to replace old ones. But cancer throws a wrench in that system. Collins, Jack, and Pavlich explained this beautifully in their research back in 1997. Their work was some of the earliest to detail how mutations in genes like P53 and RB1 lead to unchecked cell growth. Even though their study is over 25 years old, their findings are still the foundation of our understanding of cancer biology today. They described P53 as the guardian of the genome, a protein that ensures cells with damaged DNA either repair themselves or self destruct. Without P53, damaged cells slip through the cracks, divide uncontrollably, and they become tumors. Now, this breakdown doesn't just happen randomly. Cancer cells are master manipulators. Hernandez Borrero and L. Dairy, more recent works build on this by showing how different mutations in P53 contribute to specific types of cancer. So, for example, in breast cancer, mutations in BRCA1 and overreaction of CDK4 and 6 create a kind of domino effect, allowing cells to bypass the G1 checkpoint. The combined knowledge from other studies like Collins and newer research by Hernandez Borrero and El Dairy show us that while our understanding of cancer has evolved, the core principles of cell regulation remain critical in fighting the disease. Now, if you think cancer cells are just chaotic, think again. They're actually master tacticians with strategies that would make a supervillain proud. One of the most fascinating things researchers have discovered is how cancer tricks the body into supporting their growth. Otto and Sinsky's research showed how cancer cells can hijack the body's normal signals to keep dividing. They overproduce growth factors, those chemical signals that tell the cell to divide. It's like they're drowning in sticky notes that all say keep going. Cancer cells, they can ignore this signal that's telling them to stop. Normally when cells they crowd together, they get the message to stop dividing. But cancer cells, they do not care about personal space. They just keep on growing and keep on forming all of these dense tumors. Now, here's another wild fact. Veslov and his team discovered that cancer cells can even manipulate their environment. So, they recruit blood vessels to feed the tumor in a process called angiogenesis. It's like they're building their own supply chain to ensure that they have enough oxygen and nutrients they need to thrive. Now, let's get down to two key players in this story, MYC and P53. MYC is what scientists call an oncogene. In normal cells, MYC is like a helpful cheerleader, promoting growth and division when it's actually needed, like during development or healing. But in cancer cells, MYC turns into a reckless drill sergeant, constantly pushing the cells to divide, even when it's not safe. It's like putting the cell's cycle's accelerator pedal to the floor and snapping the brake line. On the other hand, P53 is a tumor suppressor. You could think of it as the cell's wise guardian or quality control inspector. When P53 is functioning properly, it can pause the cell cycle to repair damaged DNA. Or, if the damage is too severe, it orders the cell to self-destruct to protect the body. But in cancer, mutations often render P53 useless, so the inspector has been fired. Without P53, there is no one to catch damaged cells, and they're just free to multiply and spread. Even though Collins and Jack and Pavlich's study is decades old, Their description of these processes, how oncogenes like MY3 turn on the gas pedal and the tumor suppressors like P53 fail to hit the brakes, remains fundamental. It's amazing to see how newer studies have expanded on their work, adding layers of detail about the molecular tricks cancer uses to survive and grow. Okay, guys, so we've covered so much ground so far. We've talked about how cancer hijacks the cell cycle, but what is being done about it? Let's now talk treatments. We're going to slow it down a little bit, guys. We have some big words of drugs, and so I want to make sure everyone's following along as we continue on the cell cycle in cancer. So, one of the most exciting breakthroughs in cancer therapy has been the development of drugs called CDK inhibitors. Let me break this down for you. CDK inhibitors, or the cisplatin-dependent kinases, are enzymes that act like a green light in the cell cycle. They signal the cell to move forward onto the next phase. In many cancers, particularly breast cancer, two of these enzymes, CDK4 and CDK6, are overactive. Think of it as a malfunctioning traffic light that keeps blinking go even when it shouldn't. Drugs like palbociclib, ribociclib, and abemaciclib are designed to block CDK4 and CDK6. These drugs are essentially hitting the brakes on the out-of-control cell division. They specifically slow down the transaction from the G1 phase, the growth phase, to the S phase, DNA synthesis phase of the cell cycle. It buys time for the damaged cells to either repair themselves or self-destruct. It's like throwing a wrench into a runaway assembly line to stop the production of the defective products. Okay, let's talk about some results. So Otto and Siskinsky, they highlighted an effectiveness for these drugs, particularly in the hormone receptive positive breast cancer. This is a type of breast cancer where the tumor cells have receptors for hormones like estrogen and proestrogen, which fuel their growth. So by targeting the CDK4 and the CDK6, patients that have this type of cancer, they have had significant improvements in progression-free survival rates, basically meaning that they have been able to live longer without their cancer worsening. But these innovations, they don't just stop there. Researchers like Veslov, his team, they were taking cancer treatments to the next level during that time with advanced drug delivery systems such as nanoparticles. What are nanoparticles? They are, imagine tiny microscopic carriers about 1,000th of the size of a strand of hair that can transport drugs directly to cancer cells. These carriers can be even designed to release their contents only when triggered by certain conditions, like the acidic environment that is inside a tumor. It is like designing a smart missile that seeks out cancer cells and spares the healthy tissue. This approach, it not only increases the effectiveness of treatments, but it also reduces the nasty side effects that also comes with like traditional chemotherapy, like nausea and hair loss and fatigue. Now, but it all sounds good, but things still get tricky. As promising as these treatments can be, or cancer cells, they're clever. Over time, they can develop drug resistance. This happens when the cancer cells evolve. They find an alternative pathway to just keep growing, even when we block out the CDKs. Think of it as the cancer taking a detour to avoid a traffic jam we just created. Siskowski emphasized this challenge and pointed out the importance of combination therapies to combat resistance. What are combination therapies, Kerstin? It's when we use more than one type of treatment at the same time. For example, pairing CDK inhibitors with immunotherapy. This will boost the body's immune system to recognize and attack cancer cells. Immunotherapy drugs like immune checkpoint inhibitors, they work by unmasking cancer cells so that the immune cells, they're able to target them more effectively. When used alongside CDK inhibitors, the hope is to make it much harder for the cancer to find a way to escape. So we've covered so much this far, and to wrap this all up, we're going to now talk about why all of this is important, because understanding the cell cycle isn't just about biology, it's about hope. For me, this research isn't just theoretical. My mom's breast cancer is driven by some of the very mechanisms that we talked about today. Her treatment plan includes therapies that target those overreactive CDKs, and learning about how these treatments work has been empowering for me. It gives me a sense of control in a situation that often feels overwhelming. But it's not just about my mom. Millions of people around the world are fighting cancer, and thanks to the work of scientists like Collins, Barrio, Vinslove, and Otto, we're making progress. We're learning how to stop cancer in its tracks and how to tailor treatments to each patient and how to turn the tide against this disease. Now, let's zoom out and tie this back into cell biology, the field where it all begins. For students that are out there who are studying biology, think of the cell cycle as the backbone of everything we're talking about. If you're learning about processes like DNA replication, checkpoints, or proteins like p53, you're studying the very foundation of how cancer develops. For example, understanding the G1 and S checkpoint isn't just a test question. It's the key to understanding how cancer cells bypass regulation and grow uncontrollably. And for those with personal experiences, whether you're supporting a loved one like I am or going through it yourself, knowing these mechanisms can help you understand what is really happening on a deeper level. The cell cycle is where cancer starts, and it's also where we can intervene. It shows us how tiny disruptions in a seemingly mundane process can snowball into something as devastating as a tumor, but it also shows us where to focus our fight. If there's one thing I want you to remember about the cell cycle in cancer, it's this: it's all about balance. The cell cycle is a carefully choreographed dance. Cyclins and CDKs regulate progression. Tumor suppressors like p53 and RB1 act like the brakes, and the checkpoints ensure everything is running smoothly. Cancer happens when this balance is lost, when the brakes fail and the gas pedal gets stuck, and the cell cycle spirals out of control. In 2024, our understanding of these processes have brought us closer than ever to targeted personalized treatments. CDK inhibitors are a prime example of how cell cycle research has directly translated into saving life therapies. And the work doesn't stop there. Scientists are now exploring immunotherapies, nanoparticles, and combination treatments to improve outcomes even further. So whether you're a student just trying to wrap your head around mitosis or someone who is deeply invested in understanding cancer for personal reasons, this knowledge is powerful. It connects the dots between what happens inside a single cell and the treatment that saves lives. And it reminds us that even the smallest processes like cell dividing can also have the biggest impacts. Before we wrap this up, we're going to talk about one of the most exciting modern treatments, the CAR T cell therapy. This approach involves removing a patient's T cells, a type of immune cell, and genetically altering them in a lab. These modified T cells are given a special receptor called a chimeric antigen receptor, which allows them to specifically target and destroy cancer cells. Now, how does this connect to the cell cycle? CAR T therapy takes advantage of the fact that the cancer cells are constantly dividing. By engineering T cells to recognize markers unique to the dividing cancer cells, this therapy turns the immune system into a precision weapon. It seeks out and destroys cells that are stuck in overdrive, effectively addressing the unregulated growth caused by the cell disruptions. While CAR T therapy has shown incredible results in blood cancer like leukemia, researchers are now working to expand and use to the solid tumors. It's yet another way that our understanding of the cell cycle and cancer biology is translating into saving life of innovations. All right, we have now come to the end of the podcast. So what have we learned today? At its core, cancer is a disease of broken regulation, a disruption of the cell cycle, that intricate process that keeps our cells growing, dividing, and dying in harmony. When the brakes fail, like with mutations in tumor suppressors like P53 or RB1, or when the gas pedal is stuck down thanks to overactive oncogenes like MYC and the balance is lost and cancer takes hold, but we are not powerless against it. Scientists have spent decades studying these mechanisms, and their work has brought us incredible breakthroughs. Treatments like CDK inhibitors, which slow down overactive enzymes like CDK4 and CDK6, are already saving lives. Advanced therapies like CAR T cell therapy are using the immune system to hunt down and destroy cancer cells, while innovations like nanoparticles are delivering treatments with pinpoint precision. What ties it all together is the cell cycle. It's not just a lesson in your biology textbook. It's the key to understanding how cancer begins. It's more importantly how we can use it to fight it. For students listening, remember that the cell cycle is more than just G1, S, G2, and the M phase. It's a story of balance and control. For those personally affected by cancer like my mom and so many others, understanding these concepts brings clarity and I hope some empowerment. Cancer is complex, but as we've seen, it's not invincible. Every discovery, whether it's about how the cells divide, how cancer tricks the body, or how we can use these treatments like immunotherapy and precision medicine, bring us closer to the future where cancer is something that we can manage or even cure. So thank you for joining me today on Kerstin's Microverse, and I hope you leave this episode with a little bit more knowledge, a lot more curiosity, and maybe even a sense of hope. If you found this episode helpful or inspiring, share it with someone who might need to hear it. Until next time, keep asking questions, stay curious, and never forget, even the smallest process, like a single cell dividing, can have the biggest impact. See you next time.
Let's go, lose control, slip and slide on the dance floor. Let's go, lose control, slip and slide on the dance floor.